Yes.
Yeah, perfect.
The topic of my talk is the use of green nanotechnology for human lung adenocarcinoma and high performance for supercapacitor application. I am Dr. Kalavati Seni from Miranda House College, University of Delhi, India. This is my college. This is a heritage building of the world. Outline of my talk is the green synthesis of zinc oxide as well as copper oxide nanoparticle, and also the synthesis of zinc oxide by chemical wet method, characterization using advanced technique, anti-cancer study on human lung cancer cell line, that is A547, electrochemical study for supercapacitor applications. These are the trees. There are two are trees. One is the neem Azeridica indica, other is the Ficus bengalensis banyan tree, and third one is the shrub, that is Taberni monetica divertica. Their leaf we have used in synthesis of the metal oxide nanoparticle. This is the recent article which in press for publishing. This is in vitro study of green synthesized zinc oxide nanoparticle by using 1 ml of Azadirachta indica leaf extract. I am going to present the result of this paper also, and other paper which is regarding 2 ml of Azadirachta indica extract by using uh, extract of this tree, we have synthesized zinc oxide nanoparticle, and we have compared the result by using the zinc oxide nanoparticle which is synthesized by using chemical wet method. This is the graphical representation of my comparative study by zinc oxide synthesized by green method as well as by chemical wet method. In green synthesis, we have taken 1 ml in one set of experiment, and in another set of experiment, we have taken 2 ml of leaves extract, uh, 2.2 molar zinc sulfate, and zero point 0.2 uh, molar sodium hydroxide, and put it for 2 hours at 40 degrees centigrade at hot magnetic stirrer. In this case, we have obtained rod shaped zinc oxide nanoparticle. These are the FTR, powder XRD, UV visible, and FESM of these synthesized green particles. And these particles have been used for anti-cancer study, in vitro study of lung cell line, and these are the apoptosis of the cell line. And when we have used the same particle by synthesize chemical wet method, in which we have used 200 millimole trisodium citrate, 1 gram sodium hydroxide, and zero point 0.2 molar of zinc sulfate, then we the powder oxide is here, and FTR also is here, and UV is also here, and the morphology is this, but this particle doesn't show any induced apoptosis. Why we use green synthesis? Because it is preferred over traditional synthesis. Phytochemical present in plants serve the role of both reducing as well as capping agent. It is very cheap, easier, non-toxic, fast and safe method. The phytochemical present in the extract have the functional group like OH, CO, amines, and many more to enhance the targeting properties. Why we have used the only neem leaves extract or Azadirachta indica extract? Because these leaves consist of 40 different active compounds called terpenoids or specifically liminoids. These phytochemical have the property like antifungal, antidiabetic, antibacterial, antiviral, anti-cancer, and sedative properties also. Not only leaves, other part of these tree like seeds, bark, also seeds, bark, peels of the fruit also are very effective for different type of the application like antiseptic, antiviral, antipyretic, inflammatory, anti-ulcer, antifungal, as well as anti-cancer also. And these are the specific phytochemical present in the Azadirachta indica, that is azadirachtin, nimbin, nimbidine, nimbidol, sodium nimbinate, quercetin also. Why zinc oxide nanoparticle? Because it is very less toxic, stable, and it is n-type semiconductor, shows various properties like antibacterial, antidiabetic, anti-cancer, as well as supercapacitor. The green synthesized nanoparticle by our research group are used for antibacterial application, anti-diabetic also. Very recently, we have communicated one paper for anti-diabetic properties, and the band gap we have obtained 2.4 electron volt to 3.146 electron volt. The synthesis I have already mentioned. We take the 25% aqueous extract of leaves of Azadirachta indica and take 50 ml of 2. A molar zinc sulfate heptahydrate, 1 ml aqueous extract of leaves, that is regarding 25%. And after 5 minutes, we have added 50 ml of 2 molar sodium hydroxide and stirrer on hot magnetic stirrer at 40 degrees centigrade for 2 hours. Then we get the white colored precipitate as well as in colloidal state the particles. Then we dry and wash and dry in hot air oven and then use for other characterization. These are the UV. The lambda max is obtained 370.01 nanometer. It is regarding 1 ml of leaf extract and it is the band gap 2.5 electron volt for the green synthesized zinc oxide nanoparticle regarding 1 ml extract 25% of Azadirachta leaves. These are the powder XRD pattern which match with the JCPDS number 1451. And this is the uh, FTR spectra which shows incorporation of various functional group which is coming from phytochemicals present in the extract which are by which the particle is coated or we say green coated. And these are the three comparative powder XRD. This XRD is regarding Ficus bengalensis and this is for other plant that is Jessamine Ravani monatica and this is regarding 1 ml of leaves extract Azadirachta indica. All three uh, powder XRD are zinc oxide which is uh, related to green methods. And this is the comparative study regarding 2 ml of leaf extract when used in synthesis of zinc oxide and Zn nanoparticle is the nanoparticle which is synthesized by using chemical wet method in which we have used 200 millimole of trisodium citrate, 1 gram sodium hydroxide and zero point 0.2 molar zinc sulfate heptahydrate. These are the lambda mix and for green synthesized zinc nanoparticle for L2, it means 2 ml extract with the band gap is 2.46 electron volt and the zinc oxide synthesized by chemical wet method shows little bit higher band gap that is 3.146 electron volt. These are the uh, FTR spectra. Red one is regarding only extract of Azadirachta indica leaves. This is blue one regarding FTR for zinc oxide nanoparticle synthesized by chemical wet method, and third one that is regarding L2 nanoparticle which is synthesized by using 2 ml of leaf extract. And these are the comparative powder XRD of zinc oxide. Red one is for leaves extract and blue one is the zinc oxide nanoparticle when synthesized by using chemical wet method. There are two peak 012 and 131 and also 032. These are the regarding impurity that is coming from sulfate which is remain unreacted during the chemical wet method. It means the synthesized green nanoparticle are more pure. These are the same FESM images regarding green synthesized nanoparticle L2 with 2 ml of leaves extract. This is the TCA gravimetry analysis, and these are the same images regarding zinc oxide nanoparticle with chemical wet method. The here is first is sharp mass loss. The first mass loss is seen at 33.5 degree to 101.56 degree centigrade, which is due to presence of absorbed water on H2O. When it is heated, then first of all that H2O which is absorbed on its surface is evaporated, and second mass loss is 101 to uh, 0.56 degree centigrade to 425 degree centigrade, and the in this there is the one, this one, and then this one. There are two mass laws. The second mass law and first, all these mass law are the this area due to decomposition of phytochemical which are present in coating of zinc oxide particle. In last one, this is the degradation of zinc dihydroxide take place around 300 degrees centigrade, and after that the remaining mass loss is occurring from 425 degrees centigrade to 941 degrees centigrade, that is continuously. And last, if we again continue, then we get only zinc metal if temperature gravimetry analysis is done in presence of nitrogen gas. And if it is done in presence of air, then last one is only zinc oxide remain. The uh, size of L2 nanoparticles is width is 50 to 60 nanometer and length is 200 nanometer to 600 nanometer. And for chemical weight method, the width is 75 to 125 nanometer and the length is ranges zero point 0.5 microam to 1 microam. It means with chemical weight method, the particle have higher dimension than the green one. These are the TEM images. First one is regarding green synthesis, second one regarding wet chemical method. It means in green method, the particle are in more agglomerated state. These are the percentage composition of zinc and oxygen, or these are the EDS spectra of synthesized nanoparticle. In green synthesized nanoparticle, the zinc percent is 51.42% and oxygen 48.58%. And uh, for chemical wet method, the zinc percent is a little bit high, 52.02%, and the oxygen is 47.52%. 0.98%. These are the zeta potential study. Zeta potential regarding green is minus 28.64 millivolt, and for chemical method 34.75 millivolt. It means both are stable particles. These are the results regarding L2 nanoparticles. Uh, these are the MTT assay. Herein, the concentration is ranging uh, from 100 to 250 microgram per ml. Control one is only PBS is given in the cell line, uh, and this one, the study regarding with chemical wet method particles, not only the zinc oxide particle with green leaves extract, only extract also act as anti-cancer, and these are the value of P regarding MTTSA, which shows anti-cancer activity. And these are results regarding 48-hour incubation period. These are the comparative uh, band gap regarding Tabarni Montana. This is already I have discussed with L1, and this is L3. I have prepared one more zinc oxide with 3 ml of Tabarni Monatica. And these are the FESM and EDUX regarding L1 nanoparticle, means 1 ml extract when used in synthesis of zinc oxide particle. Herein again shows the purity of synthesized nanoparticles. These are the percentage ratio regarding oxygen and zinc. No other element is found. And these are the FESM images of zinc oxide when it is synthesized with leaf extract of banyan tree. When 1 ml extract is used, there is 10% extract is taken. And these are the FTR spectra. And these are the uh, morphology of cell line, lung cell line when treated with 1 ml of zinc oxide when synthesized with 1 ml leaf extract. This is control one. This is B cell treated with zinc oxide with chemical wet method. C is when treated with only extract of leaves, and D when treated with zinc oxide which are synthesized with leaf extract, and the IC50 value is 138.5 microgram per ml. And these are the uh, again morphology of lung cell line when treated with uh, zinc oxide which is prepared with 2 ml extract of Azadirachta indica. This is control one, and this is again the zinc oxide which is synthesized with chemical wet method, and it is with extract only, and this is the images when treated with zinc oxide with 2 ml of leaf extract. These picture are taken with Nikon uh, microscope at 200 uh, magnification. And these are the morphology and induce apoptosis analysis of A549 cell. Double strand has been done with uh, AO and propanium isopropoxide under the fluorescent microscope, brand Nikon at 200x magnification. This picture regarding the zinc oxide particle when treated with when synthesized with 2 ml, and with 2 ml it is clear the IC50 value is less than the L1 nanoparticle. It means when the extract amount is increased, the anti-cancer activity of particle is increasing, or we say the efficiency to kill the cancer cell line is increasing. And this is control one. This is with chemical wet method picture, and red one is the killed cell. Uh, EA early stage of as in secondary necrosis, early apoptosis, secondary necrosis. All these are the uh, stages of induced apoptosis. This is the quantitative result regarding apoptosis uh, has been performed with help of flow cytometry, and the G2 the percent of G2 phase is found 22.6%, and S1 phase 13.4%, for G1 phase 58.7%, and this is the B cell treated with zinc oxide when chemical wet method, this one result, and C1 when treated only with extract of leaves of Ageridica indica, and D when this one when uh, cell are treated with zinc oxide which has been synthesized with help of 2 ml of leaf extract of Ageridica indica. These are the again flow cytometry analysis regarding L1 nanoparticle when synthesized with 1 ml of leaf extract of Ageridica indica. Again, the same things here is given. A shows the untreated cell or control group of the cell. B cell treated with zinc oxide which has been synthesized with chemical wet method. C with only extract, and D with zinc oxide particle which has been synthesized with 1 ml of leaf extract. And these are the conclusions regarding anti-cancer activity. One thing is there, the IC50 value for zinc oxide nanoparticle is 138.54 microgram per ml after 48-hour incubation, and IC50 value for 2 ml of leaves extract, the particle which are synthesized with this 2 ml extract is 130.95, which shows the anti-cancer activity is increasing. Here, herein I have only discussed with 2 nanoparticle regarding 1 and 2 ml. We have also performed with other variation of volume of leaf extract. We have found the anti-cancer activity is increasing and IC50 value are decreasing. And these are the percentage regarding induced apoptosis, and or we say the result of uh, flow cytometry. Both L1 nanoparticle and only extract treatment in A549 cells have arrested cell cycle in G1 phase, S phase, and G2 phase, but the G1 phase cell percentage has been found higher, that is 83.1% when treated with rod shaped L2 nanoparticle than treated with only extract of leaves of Ageridica indica, that is 79.79%. The neem leaves extract also showed apoptosis by chromatin condensation, cell shrinkage, nuclear or cytoplasmic fragmentation in A549 lung cell line. Our result indicate that advanced experiment research on lung cancer cell must be considered for the determination of universal mechanism in the anti-cancer activity of rod-shaped zinc oxide nanoparticles. Now, the result regarding supercapacitor application. This paper also in press where we have synthesized zinc oxide and as well as copper oxide nanoparticle using 1 ml of leaves extract of Ficus bengalensis, where we have used only 10% aqueous extract, and also the supercapacitor application regarding Tabani monatina divertica, where 3 ml leaves extract have been used. Like leaves extract with Azadir indica, zinc oxide are multifunction. In the same way, the particle which are synthesized with help of banyan tree leaves extract also multifunctional. Property shows we have studied antibacterial, photocatalytic, and supercapacitor application. These two, anti-cancer and photovoltaic application, yet has to be studied. These are the theory part which I am skipping. The synthesis I have already mentioned, like Azadir indica extract, just we have replaced the extract 10% and 1 ml with only 1 ml. Uh, I have discussing here, though we have synthesized with various volume, but herein I am discussing with only 1 ml extract results. Uh, these are the leaves. In the same manner, the quantity of zinc precursor, the zinc sulfate heptahydrate is taken 0.2 molar, 1 ml leaf extract having 10%, 2 molar NaOH, and again with the stirring at a 4 magnetic stirrer for 2 hours, the synthesis is done, and the particles are precipitated and some are in colloidal state. These particles are washed with uh, 95% alcohol, then dried, and then further characterization done. This is the schematic diagram. These are the F FESM images of zinc oxide nanoparticle is fluorine shape, and the copper oxide is bigger sheet shape, as well as few smaller size particles are obtained. These are the ERAX result. This one regarding zinc oxide, and next one is for copper oxide. These are the atomic percent ratio which we have obtained by EDSS. These are the powder XRD. Uh, Herein, the crystallinity of a zinc oxide particle is higher than the copper oxide, which is uh, synthesized in the same manner, but crystallinity is higher for zinc oxide particle. And these are the JCPDS number for zinc oxide 361451, and for copper oxide JCPDS number is 89589. These are the FTR spectra, which shows the presence of phytochemical in the zinc oxide as well as in copper oxide nanoparticles. This is the comparative FTR spectra. These are the characteristic transmittance band regarding both green synthesized zinc as well as copper oxide native particle. These are the specific group which are present in their coating. And the electrochemical study has been done by using cyclic voltammetry technique, where 0.5 molar sodium bicarbonate used as a supporting electrolyte. 3 electrode system has been used. Platinum used as a working electrode. Drop casted platinum. The synthesized green particle are drop casted on platinum, that is working as a working electrode, AgAgCl as a reference, and platinum counter electrode. And the different scan rate at which cyclo uh, voltammogrammer scan. These are the CV. This is regarding zinc oxide and this is regarding copper oxide. And these are the formula regarding specific capacitance, energy density, and this is power density. S is the voltage scan rate, M is the mass of the electrode material, and V2 minus V1 is the window, potential window, which we use during scanning of cyclic voltammetry. And here P is the power density, E is the energy density, which is uh, calculated by this formula, and T is the time taken in charge discharge process. These are the results regarding first table regarding zinc oxide and this is regarding copper oxide. The energy density is higher than the for zinc oxide than the green synthesized copper oxide. These are the curve regarding the specific capacitance versus scan rate. When scan rate is increasing, the specific capacitance is decreases because at higher scan rate, the kinetic energy of electron is high, so they have the less tendency to sit down or to stress. So, uh, capacitance, if we want uh, good capacitor, so we should try at a lower scan rate. These are the summary regarding supercapacitor study or cyclic voltammetry study regarding green synthesized nanoparticles. FTR shows the presence of phytochemical which is coming from leaves extract, and already I have mentioned the higher capacitances with respect to zinc oxide. Again, third one is the result regarding the zinc oxide which has been synthesized by using 3 ml extract of Taberni montana diuretica. This is also uh, already published result. Uh, all things or experimental parameter is taken same except 3 ml extract of this leaf extract is taken and the 25% aqueous extract here has been used. These are the powder XRD and UV analysis of these synthesized particles. These are the SCM images. These are again rod shaped and bigger particles having bigger dimensions. And this is the percentage ratio of zinc as well as oxygen. Zinc is 46.74% and oxygen is 53.2%. These are the cyclic voltammetric curve or voltammogram regarding zinc oxide with 3 ml of this mean extract, and this is the specific capacitance versus scan rate curve. Herein we have uh, used uh, uh, in the first case the working electrode was the platinum, drop casted platinum. Herein the working electrode is glassy carbon electrode. We have modified glassy carbon electrode by using the uh, particle which are drop casted, its circular part which present in bottom, and AgAgCl is a reference electrode, and platinum mass was used as a counter electrode. When we compare the result regarding zinc oxide with Ficus bengalensis and Taberni montana, then we see the higher capacitance are obtained with respect to these particles, and the energy density as well as power density are very much high than the banyan tree. It means the zinc oxide which synthesize with Tabani-Montana are more good material for making the modified electrode or for a material for battery storage. These are the galvanostatic charge discharge curve when zinc oxide, rod shaped zinc oxide are drop casted on glassy carbon electrode, and this silver chloride electrode is used as a reference electrode, and these are the different 10 cycle uh, with respect to AgAgCl electrode, and time is taken 6000 seconds. We can summarize the whole result like comparative in vitro study of green synthesized zinc oxide with L1 particle as well as L2 particle. Both green synthesized zinc oxide with Azadirachta indica shows that it can be used as a nanomedicine for human lung cancer in the future. The FTR study shows the presence of leaves extract of in the particle synthesized green particle like Azadirachta, Ficus bengalensis as well as Tabani montana. All phytochemical are confirmed which are present in the coating of these green synthesized nanoparticle, and presence of these green phytochemicals, the anti-cancer property are shown. As we have already mentioned, the chemical wet method synthesized zinc oxide doesn't work as a anti-cancer agent, and the zinc oxide particle which synthesized with 1 ml of Ficus bengalensis and 3 ml of Tabani montana both can be used as a electrode material for high performance of supercapacitor application in the future. When we compare both zinc oxide with the different uh, phytochemicals out of here discussed, the zinc oxide which is synthesized with help of Tabani montana is more best as the electrode material than the Ficus bengalensis. These are my research group, and I acknowledge my research students Sapna Yadav, Mona Seni, my colleague Niti Bhatia, and these one student also, Nutan Rani, which is not here, and these are the other students. I also acknowledge to my principal, Miranda House, Dr. Vajalakshmi Nanda, for providing the research lab, and also funding agency, uh, Council of Scientific Research and Industrial Research. These are the reference number. Thank you.